Everyone, and welcome to Module 7. Last week we were introduced to lipids and we talked about their digestion, absorption, and transport. And this week is our kind of big metabolism week. Next week we'll talk about essential fatty acids, and the week after that I won't have a lecture, we'll just have readings, and we're going to start examining some of these changes and guidelines to saturated fats and cholesterol. So this is kind of the heaviest week for lipids. So let's go ahead and get started. So our objectives are to explain fatty acid oxidation and the role of carnitine in fatty acid transports, discuss the mechanisms of ketogenesis and why it's clinically significant, and also explain fatty acid and triglyceride synthesis. Okay, so last week we talked about how fat is transported to our different tissues. Now we're going to take it to the next step and talk about how fatty acids are actually broken down for energy. Now, first, fatty acids have to be activated in the cytoplasm before they can be oxidized in the mitochondria. And this might sound kind of familiar. So remember back to carbohydrates when we had to have the glucose that was phosphorylated by glucokinase in the cytoplasm in order to be activated as well. So this activation in fatty acids is catalyzed by fatty acyl-CoA ligase or acyl-CoA synthetase. It has all these different names just to confuse you. And this activation does require 2 ATP. So let's look at this. So first we have our fatty acid. It's combined with CoA, undergoes phosphorylation through acyl-CoA synthase, or we could also see that it's also called a thiokinase. Which explains the phosphorylation. And you may only see 1 ATP here, but the other ATP is used to hydrolyze this diphosphate here in order to ensure that this process doesn't go backwards. And then in the end, we have our fatty acyl-CoA, which is our activated fatty acid. Okay, now here is a figure that shows how fatty acids are transported into the mitochondria for oxidation. Okay, so I'm just going to kind of walk through this process here. So long-chain fatty acids and their CoA derivatives cannot cross into the inner mitochondrial membrane. They can only cross into the outer mitochondrial membrane. So this requires a transport system. Okay, so transport of fatty acyl-CoA into the mitochondria is accomplished via acyl-CoA carnitine intermediate. Okay, so here we see we have our free fatty acid and our ATP. This is just what we talked about, that fatty acids being converted to fatty acyl-CoA with our AMP. And this fatty acyl-CoA is able to transport through the outer mitochondrial membrane. At this point, it needs to combine with our carnitine to get this carnitine intermediate. And it does this with the enzyme here, carnitine palmitoyl transferase 1. That's a mouthful. And then the carnitine molecule right here can be transported into the mitochondria by action of this enzyme, carnitine acylcarnitine translocase. So this acylcarnitine can come into the inner mitochondria. And so now we are in the inner mitochondria where we can actually undergo beta oxidation, okay? So then this carnitine, then this acylcarnitine undergoes kind of a splitting really, okay? Where the CoA is separated from, where the CoA combines with the acylcarnitine. The carnitine is separated and goes back into this kind of intermembrane space. It's separated by this enzyme here, carnitine palmitoyl transferase 2. So we're left with the carnitine that goes back into the intermembrane space and the acyl-CoA, which can then go straight into beta oxidation to be broken up for energy. Okay, so what is beta oxidation? Well, it's basically how we break up fatty acids in order to utilize them through energy. And what occurs on the most general level is that it's a removal of 2 carbon units of oxidation from the fatty acetyl-CoA molecule at a time. So basically in beta oxidation, we're going to take 2 carbons off of the end of the fatty acid. Then we're going to go through and we're going to do it again where we're going to take 2 carbons off. And you'll see why this will make sense. Well, right now. So the reason that this makes sense is because these 2 carbon units that are taken from the end of fatty acids are actually acetyl-CoA. And what do we know about acetyl-CoA? Well, we know that it's one of the constituents we need to get the TCA cycle started. So one at a time, these acetyl-CoA molecules are lysed off of the fatty acid until the fat is completely oxidized. And we have acetyl-CoAs that can go through the TCA cycle. Okay, so here's a diagram demonstrating beta oxidation. So as shown here, the beta for each 2 carbon unit that's lysed off, we have 4 different steps. So first, and as I walk through, I'm going to kind of point out some of the things that hopefully will sound familiar for you. So here we're showing that our initial fatty acid is palmitoyl-CoA, which is kind of our most common 16 carbon fatty acid, okay? And then in the first step, we undergo oxidation. Well, we're oxidating the palmitoyl-CoA, but we're reducing FAD from FAD to FADH2. So this should seem really familiar. We're making an electron carrier here. And just like we talked about, it's a dehydrogenase enzyme. In the next step, we have hydration, where H2O is added. In the next step, again, we have the oxidation of our substrate, beta-hydroxyacetyl-CoA, in order to reduce NAD to NADH. So again, we have another electron carrier here, another dehydrogenase enzyme. And then in our last step, we have a cleavage, where we're cleaving this acetyl-CoA unit from the rest of the fatty acid. And you can see here that instead of 16 carbons now, we have 14 carbons. And this 14 carbon unit can go back to the beginning and undergo this whole process again. And then at that point, it will have 12 carbons, and so on and so forth. So how much energy do we get from fatty acids? Well, think about each lysing of each 2 carbon acetyl-CoA as kind of a round of beta oxidation, okay? So in this example, we have 16 carbons, and so we're going to go through 7 rounds of beta oxidation. And hopefully, let me see if I can. Okay, I'm just going to skip ahead just for a second to kind of demonstrate this. But see, after each carbon, we would have kind of a lysing spot. 2 carbons, a lysing spot. And so if you kind of write out how many rounds of beta oxidation that would be for a 16 carbon, it would be 7 rounds of beta oxidation. Because if you think about it, the last round is going to give you 2 acetyl-CoAs when it lyses those final, in the end, we'll have 4 carbons, and then that only takes 1 round of beta oxidation, then you're left with the other set of 2 carbons or acetyl-CoA. So in order to undergo complete beta oxidation, we need to go through 7 rounds of beta oxidation. And each step of the beta oxidation is going to give us 1 FADH. We see that here. It's going to give us 1 NADH. And then it's going to give us 8 acetyl-CoAs, because remember, we got 7 acetyl-CoAs from each round of beta oxidation, and then we have the last one that's left over. Okay, so this is just a repeat. For each one, we get 1 NADH, we get 1 FADH2, and 1 acetyl-CoA, but then plus 1 acetyl-CoA at the end. Okay. And then the acetyl-CoA enters the TCA cycle. So for each acetyl-CoA that we produce through beta oxidation, we then, in addition to this initial round of beta oxidation, we get these this energy from the TCA cycle in the form of 3 NADH, 1 FADH2, and 1 ATP. And that's just a review of everything we talked about with the TCA cycle. Okay, so how many ATPs would we get from a fatty acid? It's kind of a math problem, basically. So let's walk through it. Okay, so in this example, we have an 18 carbon fatty acid. And you can see that for each of these kind of slash marks represents a round of beta oxidation. So we see that we get 8 turns of beta oxidation for an 18 carbon fatty acid. So we have eight rounds of beta oxidation, 5 ATP per turn of beta oxidation. So that's the equivalent of one FADH2 and one NADH. And of course, we know we're not directly getting ATP. Those then have to go be reduced as well. Or sorry, those then need to go through the electron transport chain. And then we also but it's equivalent to 40 ATP because we have 8 rounds of beta oxidation, 5 ATP equivalents per round, so equivalent of 40 ATP. And then we also get our 9 moles of acetyl-CoA, 1 from each round of beta oxidation plus an extra. So we have 9 acetyl-CoA, and we get 12 ATP per turn of the TCA cycle. So for that part of the oxidation, we get 108 ATP. And then, of course, activating the fatty acid uses 2 ATP. So all in all, we would get 146 ATP from the complete oxidation of an 18 carbon fatty acid. Okay, now fatty acids yield, as we talked about last week, we know that fatty acids are more energetically dense than carbohydrates are. And we can see that reflected in the amount of ATP that they produce per equivalent carbon atoms, okay? So oxidation of 1 mole of 18 carbon fatty acid, as we just demonstrated, will produce 146 ATP, but oxidation of an equivalent glucose carbon atoms would only produce 114 ATP. So we see that we can get more energy from a fatty acid than from a carbohydrate. Now, the example that I've given you of how to see how many ATPs that you get from a fatty acid is pretty standard because most of the fatty acids that occur naturally are an even number of carbons. However, some of fatty acids that we see do come with odd number of carbons, and we have a slightly different way for counting how many ATPs that we get from these fatty acids. So just like an even number of carbons, again, beta oxidation is going to go through and remove 2 carbon units in the form of acetyl-CoA at a time. But then at the end, we'll have a 3 carbon unit left, which is called propionyl-CoA. And I don't know if this sounds familiar to you, but remember, we saw that propionyl-CoA or propionate could also serve as a precursor for gluconeogenesis. And why is that? Well, it's because that propionyl-CoA can be ultimately converted to succinyl-CoA. And remember that that is one of our intermediates of the TCA cycle. Okay, so how do we understand how many ATPs we get from an odd number of carbon? Well, so this is the example of C21. So it's almost exactly the same as the C18 because we just have this last 3 carbon unit here. So again, so in this we'll have 9 turns of beta oxidation. We'll have 9 moles of acetyl-CoA, but then we're left with this last propionyl-CoA that has 3 carbon units. Okay. And this, in that process, if we go back one, we see, the process of converting propionyl-CoA to succinyl-CoA does consume an ATP, but in the TCA cycle, succinyl-CoA produces 6 ATP, but then we also use 2 ATP for the activation of fatty acetyl-CoA. So walk through this, make sure you understand, but in the end, for a 21 carbon fatty acid, we would get 156 ATP. Okay. Now, a lot of so this should say clinical significance of fatty acid metabolism. Now, a majority of the clinical problems we see related to fatty acid metabolism are associated with the oxidation process. So this could be deficiencies in any of our carnitine transport, in carnitine itself, or our carnitine transport enzymes, so that the fatty acid cannot get into the mitochondria to be oxidated. And it can also be due to deficiencies in our dehydrogenases in the beta oxidation system. So deficiencies in carnitine, just like we would see from the figure and would make sense, can lead to an inability to transport our fatty acids into the mitochondria. And this issue we do see this issue in newborn infants, as well as patients undergoing hemodialysis. And symptoms can range from mild muscle cramping to severe weakness to even death, but it is easily treated by simply consuming carnitine. Okay, now we're going to move on to the process of ketogenesis, which is all related to this. Now, during periods of high fatty acid oxidation, primarily in the liver, large amounts of acetyl CoA are generated. That makes a lot of sense, right? We know our main product of fatty acids are acetyl CoA. Now, when we have so much fatty acid oxidation, then we're going to build up a lot of this acetyl CoA. And remember that when the acetyl CoA goes into the TCA cycle, it's not entering it alone, it's a cycle. So it needs to combine with oxaloacetate in order to make the TCA cycle keep going. But when we're building up so much acetyl CoA and we don't have enough oxaloacetate, then we exceed the capacity of the TCA cycle. And this acetyl CoA goes down a different pathway. And the pathway is to transform into these ketone bodies. The three main ketone bodies are acetoacetate, beta hydroxybutyrate, and acetone. So here's the process by which this happens, okay? So first we have the formation of acetoacetyl-CoA, which occurs through the condensation of two acetyl-CoA molecules from fatty acid oxidation. So here's our product here. And this is through kind of a reverse thiolase reaction of the fatty acid oxidation. So we saw that one of our enzymes that we used, just kind of want to tie this together here in order to activate our fatty acid is the thiokinase. So this is a thiolase reaction as well. So we have the combination of two acetyl-CoAs to make acetoacetyl-CoA. And then in the second step, acetoacetyl-CoA plus an additional acetyl-CoA produce our HMG-CoA, or hydroxymethyl-glutyl-CoA, commonly known as HMG-CoA. And then this HMG-CoA can either, we'll see later in the lecture, that this HMG-CoA can undergo a process to cholesterol biosynthesis. So just kind of keep that in mind, that we go through this first step to get to the HMG-CoA, which we then can use for cholesterol synthesis. But in this process, it encounters the enzyme HMG-CoA lyase, and one of the acetyl-CoAs is kicked off, so that we have acetoacetate, and this is one of our ketone bodies. And that can also be converted to another ketone body, acetone, or another ketone body, beta hydroxybutyrate. Okay, so in the early stages of starvation, when our last remnants of fat are oxidized, then the heart and muscle can consume primarily ketone bodies. And this is done so that our body, in its wisdom, can preserve the glucose for our brain, which we know has the highest usage of glucose. So ketone bodies can be utilized for energy through conversion of the beta hydroxybutyrate ketone into acetoacetate and acetoacetyl-CoA. So we can see that here, where we're in the muscle, we have our three ketone bodies, and then they undergo a reaction so that they're broken back into our acetyl-CoAs. And as we know, in the muscle, then these acetyl-CoAs can then go into the TCA cycle. So basically, so ketone body production does occur at a relatively low rate during normal feeding. We all have some ketone bodies kind of going throughout our bloodstream. occur to a greater extent if we had diabetes, starvation, very low carb diet, and that makes sense, right? Because when we have these conditions, then our body is starving, so we're going to start breaking down fats more and more. We're going to get this acetyl CoA to build up, but we don't have the oxaloacetate that we need from the carbohydrate in the diet. So then we're going to produce more and more ketone bodies because not only are we producing more acetyl CoA, but we don't have the ability in these conditions to have the oxaloacetate to kind of use up this acetyl CoA in the TCA cycle. So we kind of have two things going on here. But however, and in a normal physiological processes, when we have these ketone bodies, it's no big deal. And when people go through ketotic diets, very often it's not that big of a deal. We don't build up ketone bodies to this level that's going to be harmful in our body. However, ketones are acids, and if they build up to a very, very high amount, such as we see in ketoacidosis, then this can actually disturb the body's acid-base balance, although it is providing fuel during starvation. So we have kind of a disadvantage and an advantage of this ketone body formation. Okay, so when we have type 1 diabetes, again, we have ketone body production that exceeds the ability of the peripheral tissues to oxidize them. So in most people, when we would, a person who doesn't have type 1 diabetes, yeah, we're making ketone bodies. Yeah, we even may make a lot of ketone bodies if we're on a very low carb diet, or if they had type 2 diabetes. But in a person who doesn't have this pathophysiology, the ketone bodies can travel to different parts in the brain, in the body, and then they can be oxidized, such as in the muscle, the skeletal muscle, or the heart muscle. But in these conditions, type 1 diabetes, then the ketone body production exceeds the ability of the peripheral tissues to oxidize them. And therefore, we get this disruption in our pH, or this blood acidification. Now, this is dangerous because it impairs the ability of hemoglobin to bind to oxygen. And the consequence of this is that we kind of increase our respiration or try to, and this is kind of what we'll see in people who are having diabetic ketoacidosis, is this kind of rapid, shallow breathing in order to make up for this inability of hemoglobin to bind to the oxygen. Some keto acids can also cause nausea and vomiting, which will lead to fluid and electrolyte loss. And we also have serum hyperosmolarity from the high ketones, and plus the dehydration, plus the acidosis, and all of these can lead to the altered consciousness that we see in diabetic ketoacidosis. Okay, so here is a figure that's demonstrating how ketone bodies are produced and utilized. So we have our fatty acid, then we have our acetyl-CoA, it undergoes beta oxidation, but we have, if we have too many fatty acids, then we have too much acetyl-CoA, and that acetyl-CoA can get transformed to HMG-CoA, then to acetoacetate or hydroxybutyrate in our liver. Now, these ketone bodies go to the liver, okay, and we have our ketone bodies in the liver, and then they are ultimately transformed to acetyl-CoA, which can then be oxidized in the extrahepatic tissues. So the ketone bodies are coming from the liver, and then they go to the extrahepatic tissues where they can be utilized for energy, oxidized for energy. Okay, now let's switch gears here. So we talked about how fatty acids are transported in the body last week, and then we just talked about how they are broken down for energy. So let's switch gears here and talk about how we synthesize fatty acids. Now, other than our essential fatty acids that we're going to talk about next week, we can synthesize all of the other fatty acids that we need from precursors. And one of our precursors is acetyl-CoA, no surprise there, and the other is malonyl-CoA, and that can also be formed from acetyl-CoA. So in essence, we really just need this acetyl-CoA. So you can see why it's such an important molecule both in the breaking down and building up of fatty acids. And both the liver and the adipose tissue are the primary sites of fatty acid synthesis. Makes sense. And in order to produce this fatty acid and get this acetyl-CoA, we primarily utilize the acetyl-CoA from the mitochondria that we've already produced from metabolism. So we might get this acetyl-CoA from fatty acid oxidation that we just talked about. We might get it from pyruvate oxidation, which we talked about in the carbon or in the carbohydrate section, or we could get it from the degradation of an amino acid carbon skeleton, which we'll talk about when we talk about our protein sections. So, um, so we have acetyl-CoA in the mitochondria, but we're not going to make our fatty acids in the mitochondria. And we need acetyl-CoA. So we need acetyl-CoA to get into the cytosol where we're actually going to make the fatty acid. But one problem here is that acetyl-CoA can't pass through the mitochondria. So what are we going to do? We're going to transform the acetyl-CoA back into a citrate, which can move out of the mitochondria, and then back into acetyl-CoA once it's out of the mitochondria. So once the citrate goes back into the cytosol, first it's converted to acetyl-CoA, and then it also to oxaloacetate. So try to visualize this. Try to visualize the TCA cycle, okay, where we have acetyl-CoA and oxaloacetate, and they combine to form citrate, okay? So we have the citrate in the mitochondria. Now the citrate is going to move back out of the mitochondria, and it's going to split back up into acetyl-CoA that we need for fatty acid synthesis and oxaloacetate, okay? And then we convert that acetyl-CoA into our fatty acid, most frequently palmitate. And this requires malonyl-CoA. It requires our NADPH, which we'll remember from our pentose phosphate pathway and our carbohydrate metabolism. And it also requires the enzymes acetyl-CoA carboxylase and fatty acid synthase. And don't worry, I have figures for all of this. So fatty acid synthase, when we see it, keep in mind that it's composed of multiple enzymes, but we kind of refer to it as just fatty acid synthase. And then palmitate, when we talk about fatty acid synthesis, we're really talking about the synthesis of palmitate. And then from there, palmitate can be converted into other fatty acids using elongation and desaturation. And we're going to focus on those processes next week when we talk about essential fatty acids. So here is a drawing or a diagram of the pathways involved in fatty acid synthesis. So here we're in the mitochondrial matrix, remember, and we get to our citrate either through pyruvate degradation, or we've broken down the acetyl-CoA by beta oxidation. Either way, we get our citrate, and citrate can move into the cytosol where it's broken up into acetyl-CoA or oxaloacetate, okay? Oxaloacetate is then converted into malate, and then malate to pyruvate. But in this process, we get the NADPH production from the pentose phosphate pathway, and that can go, we need that for our fatty acid synthesis. So the acetyl-CoA plus our two enzymes here, which are, you can see up here, are acetyl-CoA carboxylase and fatty acid synthase that I just mentioned, will give us our fatty acid. So here is our model for regulation of acetyl-CoA carboxylase. So hold on, let's go back up one more time, and you can see that this is number 5 here. So we need acetyl-CoA carboxylase in order to convert acetyl-CoA to palmitate, okay? And acetyl-CoA carboxylase, or ACC, is our major site of regulation of fatty acid synthesis. So let's walk through this here. So no big surprise that we can alter acetyl-CoA carboxylase activity by phosphorylating it. Okay, so the way that we phosphorylate it is through this AMP-activated protein kinase. People also call it AMPK. And this, I have a fly here. So when we have our acetyl-CoA carboxylase that has high activity, which would mean we were undergoing fatty acid synthesis, and then we phosphorylate it, then this acetyl-CoA carboxylase ACC goes to lower activity. So let's kind of and then in order to take the phosphate group off, then we have this phosphatase here that will lead us to high activity. So you can see that if we have our glucagon that is predominating, that's going to have a negative effect on this phosphatase. And we're going to keep this ACC in low activity, which makes sense. If we have glucagon, then we're going to going to want to build up fatty acids. We're going to want to break them down for energy, right? And we can see that with insulin, it actually encourages this phosphatase, which, so if we have insulin, what our body is saying is, yeah, I want ACC to work. I want to build up fatty acids. And then we can see a similar control down here where insulin we think would increase this phosphatase, therefore keeping this kinase at a low activity and keeping this ACC at a high activity. So this is how we regulate ACC and fatty acid synthesis primarily. Okay, so we know that once we build our fatty acids, that they're stored in our cells as triglycerides in our adipocytes. And so we had our fatty acids, but we know, right, that a triglyceride is actually a glycerol with 3 fatty acids. So how do we get to that point where we're synthesizing the triglycerides? Well, this happens in the endoplasmic reticulum. And in most tissues, the major building block for triglyceride synthesis is glycerol. However, in our adipose tissue, our major building block is DHAP. Now, I have a figure and we'll walk through these. Now, the reason that we use DHAP as our primary substrate in adipose tissue is that adipocytes lack glycerol kinase. So they must have glucose oxidized to make this DHAP so that they can store fatty acids in the form of triglycerides in our adipocytes. So when we're talking about building the triglycerides, the fatty acids come from the fatty acid synthesized through the methods that I just talked about, and also through the fatty acids that are lysed off when the lipoproteins are traveling throughout the body and lysing off the fatty acids. Okay, so here is the process by which we make triglycerides in the adipose tissue, okay? So in our other tissues, remember, we're going to use glycerol as the main building block and then add on the fatty acids. But in our adipose tissue, we need this DHAP, okay? And remember that DHAP is one of our intermediates in glycolysis. So this is where we get this from. So remember in glycolysis where we had DHAP and we had G3P, and they were kind of this is where we had broken up our 6-carbon unit into 2 3-carbon units, okay? So in our other tissues, we have glycerol, and then we have glycerol kinase, so that it will phosphorylate this glycerol into G3P. But in the adipose tissue, we use DHAP from glycolysis, and that's what converts it into G3P. So at this stage now, we have our common process in all of our tissues. So at this point, we have G3P, and then we have we add a fatty acid here, okay? And so then we have a monoacylglycerol 3P. And then that's converted into phosphatidic acid. And eventually the phosphatidic acid is then converted into our 1,2-diacylglycerol. And then we add another fatty acid and we have our triacylglycerol. So I think the take-home message is our precursors are glycerol in our other tissues, including our liver, DHAP in our adipose tissue. Those are both converted to G3P. And at that point, we then add 3 fatty acid units on through acyl transferase. Makes sense, we're transferring a fatty acyl group. And then we ultimately get our triglyceride. So, how is triglyceride synthesis regulated? Well, we know that during fasting, our G3P acyltransferase here, G3P acyltransferase, is at low activity. Whereas in the fed state, especially with high carb, we have a high expression of this enzyme. So depending on our physiological state, we have different expression of this enzyme through transcript at a transcription level. Okay, and we already know that lipid lipases catalyze the release of fatty acids from triglycerides stored in the adipose tissue. So we talked about that when we talked about the fatty acid transport through the system and the lipoprotein lipase. But we have other lipases that release fatty acids in the adipose tissue, and those are listed here: triacylglycerol lipase, hormone-sensitive lipase, monoacylglycerol lipase. And the major regulator of this mobilization of stored triglycerides is our hormone sensitive lipase. And that, again, no surprise, is regulated by phosphorylation dephosphorylation. So I hope I didn't confuse you. Here we're talking about building up triglycerides, and now the triglycerides are actually in the adipose tissue. And how are we going to mobilize them? We're going to break them down. We're going to lyse them off of the triglyceride with a lipase, just like we lysed them off of the, excuse me, I have this like fruit fly, just like we lysed them off of the triglycerides in the lipoproteins that were traveling throughout the body. Okay, so here is a schematic of the role of hormones in regulation of synthesis and hydrolysis of stored triglycerides in the adipose tissue. So first, insulin promotes triglyceride synthesis by promoting glucose uptake via upregulation of GLUT4. So we talked about that a couple of weeks ago when we talked about our diabetes section, that we know that GLUT4 is our only insulin-dependent glucose transporter. But when we do have insulin and we do have high expression of this GLUT4, then we're going to get a lot of carbohydrates into the adipose tissue. And what do we see from our triglyceride synthesis? We saw that we have DHAP, which is from our carbohydrate, which is a precursor for triglyceride synthesis. Is that shown here? I don't see that in the schematic. Okay, so let's keep going. So insulin also decreases lipolysis by activating phosphodiesterase that removes CAMP, which promotes dephosphorylation and hence inactivation of hormone-sensitive lipase. So let's walk through this, okay? So here we have our glucose that's coming in through the GLUT4, and then we have our G3P, okay? So the G3P then is converted, or we can synthesize triglycerides via that. Okay, so then we have our triglyceride, right? And then we have that's stored in our adipose tissue. So now it's stored in our adipose tissue, but what are we going to do here? We're going to try to mobilize it, right? So if our triglyceride undergoes this lipase, then it's going to be broken down to our monoacylglycerol lipase, okay? So in order to do that, we need to have hormone-sensitive lipase be active, okay? Now, how do we make that active? Well, we don't make it active by insulin, because what does insulin do? What insulin is going to do is it's going to decrease our CAMP quantity. So let's talk about it from the perspective of epinephrine, actually. Okay, so what epinephrine is going to do is increase our CAMP, right? And so if we increase our CAMP, then we're going to increase our CAMP-dependent protein kinase, and we're going to keep hormone-sensitive lipase active, okay? Right. So if we're keeping our lipase active, that means that we're mobilizing our triglycerides. Does that make sense with epinephrine? Yes, it does, because we need energy. So we're going to mobilize our triglycerides. Whereas with insulin, if we have insulin, we're going to decrease our CAMP. And what's going to happen is it's going to keep this lipase in its inactive form so that we don't need to, so our body's saying, I don't need to break these triglycerides down for energy. So I want you to think back a little bit. Think to our carbohydrate section where we have glucose, okay? So what we have is we have our glucose and we have too much glucose. So the insulin is either going to try to build it up into glycogen or break it down by oxidizing it. But either way, we're trying to get rid of this glucose. Well, you can think of it as the same way for this fatty acid. So if we have the fatty acid here, if we have insulin, we can either build it up into a triglyceride or break it down by oxidizing it. But what this mobilization of triglyceride is going to do is it's actually going to make more fatty acids. Because if we mobilize our triglyceride, then we're going to have it encounter this lipase, and we're going to have more free fatty acids. Well, we don't want that with insulin. We want to decrease our fatty acids. So we're going to keep this hormone-sensitive lipase inactive. I think I covered it all. Okay, now I did just want to slip one little added figure in here, which we didn't talk about alcohol in the carbohydrate section where that pretty much it belongs. But I think where it's of the most importance for us is that we know that alcohol is a precursor for fatty acid synthesis. So, for instance, we know that alcohol can increase our ACC activity, which then increases fatty acid synthesis. And this is really one of the main reasons behind why we get this fatty accumulation in fatty liver. And here's your practice questions for you to do on your own. And now we're going to talk about cholesterol synthesis and bile acids. Now we talked some about this in our digestion, absorption, and transport talk. So hopefully this will help tie some of this together. So our objective for this section is to explain cholesterol biosynthesis and the regulation and utilization of cholesterol. We need to also, the objective is to describe the pathways and regulators of bile acid synthesis and utilization and the clinical significance of bile acid synthesis. Okay, so this is hopefully a review for you, but our main purposes of cholesterol in our body is for membrane structure, and we see that we have these cholesterols in our phospholipid bilayer for structure. And another really important role is that they're the precursor for steroid hormones and bile acids. Now, dietary cholesterol that we synthesize, dietary cholesterol and cholesterol that we endogenously synthesize are transported through circulation and lipoprotein molecules. We talked about that last week in the form of cholesterol esters. So synthesis and utilization of cholesterol has to be tightly regulated in order to prevent overaccumulation in the body, because we know that when we have too much cholesterol in our blood vessels, that this can lead to the development of atherosclerotic plaques. So let's walk through this figure from your book. So well, actually, I'm just going to let you walk through this. But basically, we know that if we have too much cholesterol, that it can be oxidized in our blood vessels, and then it leads to these foam cells, which lead to our atherosclerotic plaques. And again, hopefully this is review. Okay, so we get cholesterol from our diet, yes. And we know that the diet does have some effect on our cholesterol levels in our bodies, but maybe not to the greatest effect that we once thought. And that's what we're going to examine when we look at the literature in a couple of weeks. But we do know that at least that about half of our body's cholesterol we synthesize in our body through cholesterol biosynthesis. And this occurs mostly in the liver, but also in the intestines and other tissues. And again, just like fatty acid synthesis, it occurs in the cytoplasm with the help of enzymes that are in the cytoplasm. So we have a really similar process as we did to fatty acid synthesis. So again, The cholesterol is synthesized by by acetyl-CoA derived from the oxidation reaction in the mitochondria, again from fatty acid oxidation or pyruvate oxidation. And that's again transported to the cytoplasm, just like we talked about in fatty acid synthesis. And acetyl-CoA must be available in the cytoplasm in order for us to have cholesterol biosynthesis. Makes sense, right? Again, it enters this cytoplasm in the form of citrate, and then it's converted to oxaloacetate and acetyl-CoA via ATP citrate lyase. And again, it uses ATP. So this is pretty much the exact same thing as we saw before. But again, remember before when we had our first steps and then we said, but this goes to HMG-CoA reductase and then that goes to cholesterol biosynthesis. Well, here we are. So there's actually a many step process in cholesterol biosynthesis, but I'm just going to talk about kind of group them together and give us the main steps for the purposes of simplicity. But basically, acetyl-CoA is converted acetyl-CoA is converted to mevalonate by a series of reactions that begins with the formation of HMG-CoA. So remember, the mevalonate we needed for our fatty acid synthesis. But when we have, instead, we have two acetyl-CoA that are going to go to acetyl-acetyl-CoA, then that's going to go to HMG-CoA, and then instead of going with the process of mevalonate, instead we're going to, it looks like we can, the HMG-CoA can then go to HMG-CoA reductase, and then we're going to make squalene, and then the squalene is then converted to cholesterol. Sorry, this really isn't the best figure for that. But we saw in the other figure that, again, the HMG-CoA reductase, once the HMG encounters the HMG-CoA reductase, then we have the cholesterol biosynthesis. Sorry, what this figure is showing is that it's this process, it's this enzyme here, this HMG-CoA reductase, that most of our statins work on to block, and therefore they block cholesterol biosynthesis. Okay, so how is cholesterol biosynthesis regulated? Well, again, we know that we want to keep a constant level of cholesterol in our body, and that adults synthesize about 1 gram of cholesterol a day, but we only use 0.3 grams of cholesterol a day. So through these numbers, you can see that we really don't need to eat any cholesterol at all. We can make more than enough that our body needs. So synthesis is regulated in part by dietary cholesterol uptake or intake. And that's why we really think that the diet does have some effect, though we're not sure to what extent and how it varies in individuals. Now, both our dietary and our synthesized cholesterol are used for important functions, such as our membrane structure, steroid hormone synthesis, and bile acid synthesis. Now, we regulate our cholesterol synthesis through 3 distinct mechanisms. But the main mechanism is regulation of this enzyme HMG-CoA reductase. So we also can regulate cholesterol synthesis through our acyl-CoA cholesterol acyl transferase activity, this ACAT activity, and by regulating plasma cholesterol levels via the LDL receptor mediated uptake and HDAL mediated reverse transport. So we talked some about those last week. But right now, I'm just going to concentrate on regulation of HMG-CoA reductase. Now, we had 3 mechanisms to regulate cholesterol biosynthesis, but we also have 3 mechanisms to regulate HMG-CoA reductase. Now, we can control gene expression. We can control the rate of enzyme degradation. So we're going to control how it's made and how it's broken down. And then the main method that we're going to focus on, no surprise, is phosphorylation, dephosphorylation. And this enzyme is most active in its dephosphorylated form. And this activity is regulated by AMP-regulated protein kinase, or AMPRK is how you'll see it. Okay, so here's one of our very fun diagrams that has a lot of things going on. So let's try to work through it. Now here we see that HMG-CoA encounters HMG-CoA reductase and we get cholesterol. And that once we have cholesterol, that has negative feedback on this HMG-CoA reductase, just like we just talked about. Makes sense. Okay, so we know that we need to have this enzyme active in order to make our cholesterol. So let's walk through. I wish they had had the insulin on here, but let's walk through. So here we know that we get CAMP from glucagon and epinephrine, right? And that when we have in our second messenger system, we know that CAMP is going to stimulate our protein kinase A, okay? And we know that that is going to make this inhibitor active, okay? Which means that we're going to keep our phosphatase inactive if the inhibitor is active, and therefore we would keep HMG-CoA reductase inactive if we had glucagon and epinephrine. Makes sense, right? If we have glucagon and epinephrine predominating, we do not want to build cholesterol. We only want to build cholesterol if we have the tools to do it. So therefore, insulin would act opposite. It would have lower CAMP levels. It would not inhibit the phosphatase. And the phosphatase would be active, making HMG-CoA reductase active. And then we also have another couple of roles of this inhibitor, just like we saw in the carbohydrate section, where this inhibitor is also going to inhibit the phosphatase here, therefore keeping this AMPRK active, and therefore AMPRK active, yep. And so this is a kinase that then adds a phosphate group and makes this inactive. So again, if we had glucagon or epinephrine, it makes sense that we would want to keep this inactive. So again, we have simple processes, but a lot of them going on, which makes it complex. I think we just went through all of this, but this is in text form, so you can walk through this and make sure it makes sense. Okay, and we know that our CAMP levels are also controlled hormonally. Therefore, our cholesterol biosynthesis is controlled hormonally. So again, I just went through this. But basically, if we have glucagon and epinephrine, we're going to have high CAMP. We're going to inhibit cholesterol synthesis is the take-home message. Whereas if we have insulin, we have low CAMP, and we're going to activate cholesterol synthesis. And here, again, just like we saw in the previous figure, if we have a lot of cholesterol, that's going to increase our, not only inhibit HMG-CoA reductase, but also increase our rate of HMG-CoA reductase degradation. Okay, so how is cholesterol utilized? Well, hopefully this will be a big repeat from last week when we talked about how fatty acids are transported throughout the system and utilized. So they're transported primarily in the form of cholesterol esters on lipoproteins. Nice review. Okay, so we know that cholesterol that's synthesized in the liver and dietary cholesterol that exceed our needs in our liver are then transported into the serum via VLDLs and ultimately LDLs. And that cholesterol found on the plasma membranes can then be extracted by our HDLs and put back into cholesterol ester form by the enzyme LCAT. So, and then we know that the cholesterol is brought back to the liver and ultimately converted into bile acids in the liver, and then it can be excreted in the bile as free cholesterol or as bile salts. So again, a little review from last week. Now, the end products of our cholesterol are bile acids that we synthesize in the liver. And this is our main method for cholesterol excretion in our body. However, cholesterol excretion in the form of bile acids is not going to make up for the fact if you have a lot of excess dietary cholesterol intake. And again, we'll examine this more in a couple of weeks. But my take on it is that it's not that dietary cholesterol has no impact. If we're eating lots and lots of cholesterol, we will see the effects, but that 300 milligram level, we do not have the science to back up, is basically what my take on it is. So the most abundant bile acids are our ketodeoxycholic acid and our cholic acid. Ding, 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 ding. Does this ring a bell? Remember when we were talking about the short chain fatty acids from the carbohydrate fermentation in the large intestine, and we said that um, that the short chain fatty acids would help us shift the type of bile acids that we were producing from cholic acid to ketodeoxycholic acid. So this is just showing how the bile acids are converted to bile salts before leaving the liver. And it's basically that the carboxyl group of the bile acid is conjugated to either a glycine or a taurine before they're excreted as bile salts. So we have the two different types of bile acids here, and they're joined with a glycine or a taurine, and then we have our bile acid as salt. And we know that our bile salts are more water soluble. Therefore, it makes them effective emulsifiers, which is, of course, their entire function is to help us emulsify the fats in our lumen so that we can have better access to them, give them better access to the enzymes so that we can digest them. And after the bile salts are produced in the liver, they either flow right into the duodenum so that we can immediately use them to break down the fats in our diet, or they can be stored in the gallbladder. Okay, so 95% of the bile salts that we make are reabsorbed into the ileum. So we're going to secrete them out into the duodenum, and then nearly all of them are going to be reabsorbed into the ileum, where they'll go back to the liver, and we'll go through this whole process again. So if we look at it in terms of grams, we can see that nearly all of our bile salts are circulated throughout this sequence. So we do lose a small amount in the feces, and we do synthesize a small amount in the liver. But again, you can see when we're actually looking at the quantities that those last two are fairly insignificant compared to how we're able to recycle the bile acids in our bodies. Okay, so why is bile acid important? Well, we know that it's our only significant mechanism by which we can excrete cholesterol from the body. And again, that's in the with this little 0.5 grams loss in the feces. So it does seem kind of insignificant, but if it's really our only method of excreting cholesterol from the body, it's very physiologically important. We also know that bile acids solubilize the cholesterol in the bile, preventing cholesterol precipitation in the gallbladder. So that's really important to be able to keep this cholesterol in the form of this bile acid so that we can utilize it for digestion. The bile acids also emulsify fats in our lumen, which facilitates digestion of triglycerides by increasing their exposure to pancreatic lipase for digestion. And because they help facilitate the digestion of fats, they also help facilitate the digestion of fat soluble vitamins. And here is a little YouTube video that does a whole overview of lipoprotein and cholesterol metabolism, and I'll let you look at that one on your own. Sorry. Okay, so another reason that cholesterol is incredibly important is that we need cholesterol in order to synthesize our steroid hormones. And every steroid hormone except retinoic acid uses cholesterol as a precursor. So in this process where cholesterol is converted to our steroid hormones, we have a 27 carbon cholesterol that's then converted to an 18, 19, or 21 carbon steroid hormone involving rate limiting irreversible cleavage of a 6 carbon residue from the cholesterol to produce pregnenolone. And I have a figure here. Okay, so we have our cholesterol precursor. We then have this lysing of a 6 carbon unit to get our 21 carbon pregnenolone, and then this is then converted into all of our other physiologically important steroid hormones that have either, sorry, did I say 18 or 19 carbons. So here we have our progesterone, we have our estrone, we have our estradiol, we have our aldosterone, our cortisol. So you can see that cholesterol is a precursor for all of these really important um, steroid hormones. And I'll let you go through this on your own, but this table just walks through the importance of our steroid hormones. So let's just look at cortisol, which is our dominant glucocorticoid in humans. And that's involved in stress adaptation, elevating blood pressure and sodium uptake, and numerous effects of the immune system. So you can see that every single one of these steroids has really important physiological, is very important physiologically. And these all come from cholesterol. Now, one last topic that I wanted to talk about is that's related is plant sterols and stanols. So we as humans and animals do not produce these sterols and stanols. We only get them from the diet, and we have different forms of these sterols we can see, and we do have certain dietary patterns that have greater sterol intake, such as an Asian dietary pattern or a vegetarian dietary pattern. So why are these important? Well, why are these important? Well, these sterols and stanols have a similar structure to cholesterol, but they are slightly different in that they have a methyl or an ethyl group on them, which decreases absorbability. So we have had studies that indicate that incorporating these plant sterols into the diet could be an effective method of lowering LDL cholesterol because it inhibits cholesterol absorption. So basically, it competes with cholesterol for absorption, therefore ultimately lowering our LDL cholesterol. Now, again, we're really still trying to understand how big of a role does dietary cholesterol really have on LDL cholesterol, and to be determined, but we know that it does have some effect. And here we have just a little video that's demonstrating the mechanism here. And so again, I'll let you watch that on your own. So here are your practice questions too. And that is all I have for you today about lipid metabolism. Hopefully you felt a little bit more comfortable in comparison to the carbohydrate metabolism, because we used a lot of the common processes that we learned from that section. So next week, we're going to have a little bit shorter lecture on essential fatty acids, and I will talk to you all then.